Hello students, welcome to your third online session biology class 9th. Today we will complete following topics: Golgi apparatus, lysosomes, mitochondria, plastids and vacuoles. So our first topic is Golgi apparatus. Golgi apparatus was firstly discovered by a great scientist, Camillo Golgi. On his honor, in his honor, this particular organelle is named as Golgi apparatus. You can see the Golgi apparatus is tubular structure, flattened tubular structures, and some of the vacuoles are also here. So these are made up of some flattened tubular structures, and these tubular structures are present over here and some of the vacuoles over here. And actually, these Golgi apparatus are considered differently in the plant cell and animal cell. In plant cell, they are known as dictyosomes, and in animal cell, they are known as Golgi apparatus. They are known as Golgi apparatus. So, what is the use of these Golgi apparatus? What they do actually? They all are double membrane structure. They all are double membrane structure. And what they do, actually, whatever the protein is synthesized in rough endoplasmic reticulum, ribosomes of rough endoplasmic reticulum, which we have studied in last class, that is pinched off from rough endoplasmic reticulum and received by Golgi apparatus. Golgi apparatus modify those proteins and finally repacked, and finally after repacking dispatch it to various targets inside the cell, within the cell or outside the cell. Golgi apparatus have one more function. When they receive the protein from the endoplasmic reticulum, they modify this protein by adding some of the carbohydrate to it or some other materials. After that, they are packed. Target is already decided, and they dispatch the modified protein, dispatch in form of these vesicles to various targets within the cell or outside the cell. And one of the most major function of the Golgi apparatus is that it excuse me, many complex sugars are also made by the simple sugar in the Golgi apparatus. Okay, so as I told you that modified protein are sent to various targets within or outside the cell. So when they reside within the cell, these vessels work as lysosomes. Another cell organelle, that is the lysosomes. So now it comes to next cell organelle, that is the lysosomes. What are the lysosomes are actually? Lysosomes are the waste disposal management system of the cell. Lysosomes are considered as waste disposal management system of the cell. How it is considered like? Lysosomes are those pinched off vesicles which are reside within the cell when they are dispatched from the Golgi apparatus, and they are having lytic enzyme in it. They are having lytic enzyme in it, which is a digestive enzyme, and whenever any worn out or dead cell organelle come in contact of lysosomes, lysosome fuse with it, and its lytic enzyme digest this worn out or the dead cell organelle, and finally digestion is over. Same way, whenever any foreign particles enter into the cell, then lysosome fused with it and its digestive juice, that is considered as lytic enzyme. Lytic enzyme digests the complete foreign particle and foreign particle and break down break down all organic material or complex material into the simple material. And one more very special work is done by the lysosomes. That is what, whenever a cell during any cases or because of any reason, when cell disturb its metabolism, lysosome burst and see what happened. As lysosome is having very high digestive juice, so as lysosome burst, the digestive juice spread over the cell and it digests all the cell organelle within it. That is why they are also known as suicidal beaks of the cell. They are also known as suicide bag of the cell. Why they are called suicide bag of the cell? Because of any disturbance in cell metabolism, the lysosome burst and their lytic enzyme is spread over the cell. All the cell organelle digested by the lytic enzyme of the lysosome. So it eats up the own cell. That is why it is also known as suicide bag of the cell. Okay. So now the function of lysosome is over. It is also known as waste disposal management system of the cell. Now our next topic is mitochondria. Mitochondria, as you all know, that cell perform various functions, and that is why the cell is also considered as functional unit of the life. So to do any function, it needs some energy. So how the energy it get, or from where it gets the energy? A structure, an organelle is present, that is known as mitochondria. Mitochondria is present in the cell, and this is the structure of mitochondria. You can see it is having two membrane, outer membrane and inner membrane. Outer membrane is complete and smooth membrane, whereas inner membrane is having the finger-like projections. You can see over here, these are the few finger-like projections which are known as cristae. They are known as cristae. So these finger-like projections increase the surface area for the manufacturing of the ATP. What is this ATP? ATP is adenosine triphosphate. ATP is what? Adenosine triphosphate. Actually, what is the work of this mitochondria? Mitochondria is also known as powerhouse of the cell. It is also known as powerhouse of the cell. Why it is considered as powerhouse of the cell? To do any function, cell need some energy, and whatever the food enters into the cell, the mitochondria convert that food into the energy capsules, and these energy capsules are known as ATP. So ATP provides the energy to the cell to function, to do any function or all the chemical reactions. And as you see, these projection are present over here, and these projection helps to generate more ATPs. And a special thing of mitochondria is what? It is having its own DNA and own ribosomes over here. And the fluid which is present over here, that is called as matrix. And these projections are known as crest. These projections are known as crests over here. And the ATP is known as ATP is known as the energy capsule. And these energy capsules are present, which provide in the mitochondria. They are released by the mitochondria, which provide energy to the cell to perform various chemical reactions. And one more thing I want to tell you, that the number of mitochondria in a cell depends on the working of the organ. Means what? As you are doing the walking work through your legs, so the muscles of legs, the cells of muscles of legs are having more mitochondria, as more energy is required to move, to walk, to run. While, as you know, your ear, external ear is not moving, so there is less energy required, so less mitochondria are present in the ear cells. Is it okay? Now your fourth topic is plastids. So beta, plastids are what? The plastids are present in the plant cell only. They are not present in the animal cell. And they are of mainly two types. The first one is known as chromoplast, and the second one is known as leucoplast. So what is the difference between chromo and leuco? Leuco means, so first I am taking the leuco. So leuco means white. So white color of pigment is known as leucoplast. And chromo means color. So colored pigments are known as chromoplast. So two type of plastids are over here, chromoplast and leucoplast. Now I am taking about the leucoplast. Leucoplast are white color pigments which are present within the cell. And actually these leucoplasts are used to store the extra food, like they store the food in form of sugar, oil or starch. So the cells where white color food is stored, as you all know, the potatoes, they all are white. The radish, they all are white. So what is there? There leucoplast cells are more, which store the starch in form of extra food in form of starch over there. Is it clear? So potatoes are having more cells of the leucoplast, white cells. Another one is what? Another one is chromoplast. Chromoplast is having various pigments. They can have the chlorophyll. Chlorophyll is green color pigment. The chlorophyll is green color pigment, and the chromoplast which is having chlorophyll, they are known as chloroplast. The chromoplast which is having chlorophyll green color pigment, they are known as chloroplast. So this is the structure of chloroplast. You can see this is also double membrane structure which is having its own DNA and its own ribosomes, and the fluid which is present inside it, that is known as stroma. And these are the various circular structures are over here. These are known as granum, and these are the starch grains over here, and few of the chromoplasts are also colored plastids are also here. And this particular chlorophyll is responsible for the process of photosynthesis. This is responsible for the process of photosynthesis. This absorbs the sunlight. In addition of sunlight, with the help of water and carbon dioxide, it makes food, and this process is known as photosynthesis. And that food is used in the development of the plant, as well as extra food is stored in form of leucoplast and chloroplast. Is it clear? Now, the another type of plastid chromoplast is, that is color pigments. Colored pigments. Colored pigments means you have seen the flowers are of various colors, red, yellow, and various colors, while the green color is present in leaves, and in small plants they are of stems also. But colored pigment are present in flowers, and these colored pigments are also known as that is chromoplast. And the chromoplast gives various colors to the flowers and as well as our fruits. So I'm telling you one of the pigment, that is xanthophyll. It gives yellow color to the bell pepper or papaya. So the xanthophyll is a colored pigment which is known as chromoplast, and it gives the yellow color to the bell pepper or the papaya or such type of fruits or yellow flowers. Then it comes to leucoplast is over, chromoplast is over, chloroplast is over. Okay, beta. Now it comes to next topic, that is vacuoles. So vacuoles are what? Vacuoles are strong or storage sacs for storage of solid or liquid content. Vacuoles are small in size. Vacuoles are small in size. You can see in the book. Vacuoles are small in size and excuse me, and they store the various materials either in solid or in liquid form. In plant cells, vacuoles are full of cell sap which provide the turgidity and rigidity to the cell. What is turgidity? Means as in the daytime, when the sunlight and heat is more, the rate of transpiration increase. As rate of transpiration increase, so rate of absorption comparatively less than the transpiration. So loss of water is more than the absorption of water. So plant become wilt. And as in the evening time, the rate of transpiration reduces and a rate of absorption of mineral and water increases. That is why the turgidity pressure increases and plant become erect. And vacuoles are helpful in some of the unicellular organism like amoeba, they are taking their food using the vacuoles. As I'm showing, amoeba is having irregular structure. So this is the structure of amoeba and this is the food material. Now it takes like, it is increasing its one of the portion. These are called as, this is food material and this is called as, this is food, and these are called pseudopodia, and after that it becomes like this is food material and this is the amoeba. So it is taking its food in this way, amoeba. So vacuoles are also helpful in taking the food in the unicellular organism. In same way they excrete the waste materials over here. So beta, I'm sending you the PDF of this particular lecture. Read it, that, and learn all the topics, all the diagrams, and sending one of the worksheet with it. Solve that worksheet. Okay, bache, bye.